So welcome back friends, welcome back in another video tutorial from microbiology with Shagnik. So in that video tutorial we are going to discuss about the life cycle of Chikungunya virus. Although I am going to give you a brief introduction of chicken virus before, then we will enter inside our life cycle of Chikungunya virus. So Chikungunya virus is a positive sense single-stranded RNA genome containing virus, and their genome size is near about 11.6 kb. Okay, so remember another one thing, that Chikungunya virus belongs to family Togaviridae and genus alpha virus. Okay, and as they are alpha virus, remember one thing, that all the virus under the genus alpha virus have icosahedron capsid. Okay, along with it should have a envelope. Okay, that means the virus should be enveloped icosahedron capsid containing virus. Okay, so Chikungunya virus can found in Africa, can found in Asia and India. So let's discuss about the structure of Chikungunya virus diagrammatically. So I have told you that it is a envelope virus, and inside the envelope, I should say, rather saying inside, there will be two protein. One is E1 protein, which is on outside, and another one is E2 protein, which is on inside. Okay, and then there will be your icosahedron capsid. Okay, in green color. Then inside the icosahedron capsid you have your positive sense single stranded RNA genome of 11.6 kilobase pair in size. Okay, so Chikungunya virus, as I have told you, it is not directly transmitted from human to human, it is transmitted from mosquito, right? So the main mosquito that play a important role in the transmission of Chikungunya is Aedes aegypti. Okay, or another mosquito name is Aedes albopictus. Either Aedes aegypti, I think it should be better for remembering, and Aedes albopictus. Okay, so they can cause, that means Chikungunya virus can cause muscle pain, it can cause fatigue, it can cause rash, it can cause fever, okay, and joint pain. Okay, so that are the symptoms, and most important is acute fever can occur with temperature of 102 degree Fahrenheit. Okay, can occur. So we have discussed about symptoms, we have discussed about the structure, and now incubation period. Remember one thing, most of the Chikungunya virus infected people are symptomatic. Okay, there are two small amount of Chikungunya virus infected people, those are asymptomatic. Okay, most are symptomatic, and the incubation period is near about 3 to 7 days, with ranges from 1 to 12 days. Okay, so we have discussed a brief discussion about Chikungunya virus, what is Chikungunya virus, how they transmit, and what are the structure, what kind of genome they possess, and what is the symptoms that they can cause, what is the transmission, their mode of transmission, and what are the incubation period, right? So now let's enter in our today's main topic, that is the life cycle of Chikungunya virus. So as you can see, Chikungunya virus have receptor for their interaction with the receptor present in the surface of cell, it can be human cell. Okay, so after that interaction they will insert it inside the cell via endosome formation. Okay, as you can see, then after that inside the cytoplasm they will release the positive sense genome. Okay, so there can be two things that this positive sense genome have to complete. One is it will produce P1234 precursor, and another one is synthesis of negative sense RNA genome containing poly U repeats. Okay, so now this P1234 precursor will further undergo various steps and ultimately leads to a product that will help us, or I should say that will help the chikungunya virus to synthesize from negative strength RNA virus positive sense RNA virus. Okay, as you can see. Now this negative sense RNA virus will produce 26S subgenomic RNA, that is positive sense, and also another positive sense RNA virus containing poly A repeat. Okay, and remember that this poly A repeat containing positive sense RNA virus will in future will be assembled as the genome of the newly formed infectious virion. Okay, and the 26S subgenomic RNA positive sense subgenomic RNA will produce some structural precursors. Okay, two type of protein, C protein that will go to the formation of capsid, and PE2 and 6K and PE1 protein formation. These PE2 and PE1 are nothing but the protein formation. Those protein are present on the surface of the virus, as I have told you. That means they are E2 and E1 protein formers. Okay, then this P1 P2 P1 then this PE2 and PE1 and 6K protein usually synthesized in the ER, and then the capsid, the PE2 and 6K PE1 gene product, and the positive sense RNA genome containing poly A tail all are assembled. Okay, as you can see. So after that assembly, they will release from the cell just by lysing the cell. Okay, that means they will remove from the cell by causing cell lysis. Okay, so that is the typical chikungunya viral life cycle. Okay, so today we have learned that chikungunya virus is a virus that cannot directly transmit from human to human. Aedes aegypti and Aedes albopictus are mosquitoes that play a role of vector here. Okay, so chikungunya virus is a positive sense RNA genome of 11.6 kb in size, and it is of 100 angstrom in diameter. The entire chikungunya virus is of 100 angstrom in diameter, and chikungunya virus, as it belongs to alpha virus, it contain envelope, and it will also contain what? It will also contain icosahedron capsid. Okay, and in the envelope there will be presence of E1 and E2 protein. You have to remember. Okay, so that is the structural discussion. Then we have discussed about the symptoms that can be caused by chikungunya virus. Most important symptom is acute fever at a range of 102 degree Fahrenheit or greater than 39 degree Fahrenheit. Headache can occur, fever can occur, fatigue, rash may also occur. Okay, so that are the various kind of symptoms. Then there will be transmission. So we have discussed about Aedes aegypti and Aedes albopictus for that. Then life cycle. First they will enter by endosome formation, then they will release the positive sense single stranded RNA genome in the cytoplasm. This positive sense single stranded RNA genome will synthesize P1234 receptor, P1234 precursor, and also negative sense RNA genome. Negative sense RNA genome will contain poly U repeat, and this poly U repeat containing negative sense RNA genome will synthesize positive sense RNA with the help of a molecule that is produced by P1234 precursor. Okay, now this negative sense RNA genome containing poly U will synthesize 26S subgenomic RNA. Okay, now this 26S subgenomic RNA will leads to the formation of structural precursor, C protein and PE2 and PE1 and 6K. Okay, those protein will be synthesized in the ER, and there will be assembly of all the PE2, PE1, 6K, C and the positive sense RNA genome containing poly A repeats. Okay, so as a result, a mature virus will form and it will release by simply causing lysis of the cell. Okay, and it will also snatch some part of the cell membrane. As you can see, it possesses envelope virus and it is an envelope virus, right? And we have discussed that envelope of virus is actually not the property of virus, it is a stolen material from host cell, right? So that is the concept. Okay, so I hope this video will be helpful to you, and if it is, please hit the like button, share with your friends and subscribe to my channel to get more videos like that. So thank you for listening to this class.